essentially about ventilation and the processes associated with ventilation. So one thing that we're going to spend some time talking about now, which can take some time to wrap our heads around, because I don't particularly like the diagram your textbook uses, but I use the same diagram because it's what you guys have available to you. So I want to talk through this process and try to get us to understand exactly what's happening. But the idea here is that we have to recognize that not all of the air you breathe in, all of that fresh air, is going to be available for gas exchange. And the reason why it's not all available for gas exchange is because you have to take into account all of the structures of the respiratory tract that do not participate in gas exchange, but are still filled with air. So your trachea, the primary, secondary, tertiary, bronchi, all the way down, none of them allow for gas exchange. But all of those structures are filled with air. They're not tubes that flatten and collapse. They're not tubes that will ever be empty of air. And so that area gets called your anatomical dead space. And the reason why they call it the dead space is because it is an area of the respiratory tract filled with air, but not allowed to participate in gas exchange. So when there's fresh air in those tubes, that fresh air is more or less wasted because it can't be used for anything, okay? So we're going to kind of try to understand why it can't be used. So why is it that that fresh air that's in the trachea never makes it down into the alveoli, but instead actually goes right back out into the atmosphere? So we kind of have to disregard that volume. Generally speaking, and it's a rough estimate, your anatomical dead space is equal to your weight in pounds, so in milliliters, okay? I get asked this question almost every semester. Is it actual weight or ideal weight? Okay, what do you guys think? Ideal weight, right? Because as an individual, let's say, gains weight, your trachea doesn't get wider, your bronchi don't get wider, so you don't really change that overall anatomical dead space. So it's technically just ideal weight, okay? Now, if we were to correlate this then to pounds and milliliters, we would say that somebody who is 165 pounds would have an ideal weight again, would have a dead space of approximately 165 milliliters, right? So what that means is every time I take a breath in, 165 milliliters of fresh air is not going to help contribute to gas exchange, because that fresh air is going to stay trapped in my trachea, in the bronchi, and all the areas of the conducting zone that do not contribute to the gas exchange. So the way that we're going to represent what's happening is just by using some basic shapes that represent different parts of the respiratory tract. And understand how as air comes in and out through these areas, where that air is ultimately going, okay? So at the very top here, you have the end of your inspiration, and at the very bottom, you have the end of expiration. And we're going to kind of talk about what happens in between each of these. If you look at this key over here, it's telling you which air is fresh and which air is stale, or what we would consider kind of old air. The biggest difference between the two is their oxygen concentration. And so oxygen concentration is being depicted by P, the capital P, O2. And so here, you'll see that the fresh air is going to be the purple, or whatever's colored slightly purple. And then the stale air is going to be the kind of light blue. The stale air means it's poorly oxygenated, while the kind of purple means it's very rich in oxygen. And then what are these different shapes representing? The square represents the conducting zone. That represents your anatomical dead space. The circle represents the collective volume of all the alveoli. So essentially we have conducting zone in the square and the respiratory zone in the circle, okay? The circle is where our gas exchange is taking place, and the square is where that kind of air that really can never contribute to anything is stuck, Okay, so if we start at the end of inspiration, and what helps with this, and I know it seems silly, but don't feel like it should, because you've all been in anatomy and you've done like these things during tests. So don't feel silly doing this. Actually breathe as you think about what's happening with this, okay? So we just finished the end of inspiration. That means you just took a deep breath in, right? So as you took that breath in, you brought in some fresh air, right? Some of that fresh air came down into the alveoli, while 150 mils of it got stuck in the trachea. You're never going to see a representation of fresh air in the alveoli, because as soon as air gets down there, you have exchange going on. And so all this CO2 is getting dumped into the alveoli, oxygen is leaving, and so it just always looks like stale air, okay? So you just breathe in. There's fresh air in that trachea and all this nonsense, right? So now you're about to breathe out. When you breathe out, what happens to all that fresh air that's in the trachea and the bronchi and all of those spaces? It gets pushed out, right? Because you can't kind of exchange in the respiratory tract. You can't say, okay, I'm going to push out all this stale air, but keep the fresh air off to the side so that I can then bring that in. It's not going to work that way, okay? So you're going to breathe out, and when you breathe out, you're going to push out that fresh air, okay? Keep in mind that these tubes, this conducting zone, is not going to collapse. It's not going to flatten. It's never going to empty. So what you've done is replaced those 150 mils of fresh air that was in there with 150 mils of stale air that just came from the kind of depths of the alveoli, okay? So that means then at the end of my expiration, I have 150 mils of stale air inside of my dead zone and the normal amount of air that I would expect in the alveoli. So now, again, picture this, imagine, breathe, and think, I'm about to take another breath in, because I'm about to start my inspiratory cycle again. What happens to that 150 mils of stale air that's in the trachea, in the bronchi, that we just put in there when we breathed out? Where's it going to go? It's going to go right back in to the alveoli. For the same reasons, I can't kind of scoot aside the stale air and let all fresh air pass through. Instead, it's just going to get pushed right back down. So when you breathe in, you're going to take fresh air, about a 500 mil sample, let's say. And when that 500 mil sample comes in, your 150 of stale air goes right back into the lungs, and you only get 350 of fresh air, okay? The rest of that air that went in was stale, and now you've replaced again the dead space with fresh air. And so we're back to kind of where we started right here, okay? And so this concept of anatomical dead space and availability of fresh air for the alveoli does have important implications, okay? Most of the important implications for this are going to be associated with respiratory therapies. So when somebody's on a ventilator, or when somebody is being treated through some sort of machine. Because if you've ever observed somebody who's on a machine like that, it's not just direct into the mouth. I mean, there are tubes that go from the mouth out to this machine. So that right there is adding anatomical dead space, because that whole tube right there is going to be filled with air as it starts passing through the system. So if you give too little volume coming into the lungs, then you might not be giving enough fresh air to an individual because of all that anatomical dead space that's going to offset the fresh air coming in, okay? There is another way that I try to represent this. So this is kind of what the textbook does. I'm going to take my stab at this thing again. It's not on. Let's see. Oh, I know why it's not on. My fault. And with our ability to actually get fresh air. Okay, so you can follow the circle, square analogy if you like that one better, but drawing out the lungs sometimes helps because, again, you visualize what the actual body is doing in terms of all of these different volumes and why anatomical dead space is important. Technically, it's called deoxygenated. It isn't completely oxygen depleted. It's just low in oxygen and high in CO2. Okay, any questions on this? Yes? It mixes. So that's a good question. So does this air get more and more stale or something as we kind of go along? When the fresh air comes in and you kind of contribute, everything mixes, so it balances out to the extent where you have the same stale air all the time. The only time your stale air will change is if your respiratory conditions change. So you're in an environment maybe that doesn't have very good air circulation, and so that air is stale, and then you're breathing that in, which then makes things even more stale. So yeah, in that case, yeah. But otherwise, because everything mixes in there, it eventually just kind of balances out. Yes? So that is usually some sort of blunt force trauma that causes you to just kind of like not be able to breathe. More often than not, it is an issue with the diaphragm. The diaphragm kind of gets cramped to a certain extent because the electrical signals to it have been disrupted due to the trauma, and that causes it to not contract, and so you just kind of like gasp, but nothing happens until it kind of resets. We're going to talk towards the end of gas exchange, control of ventilation, the phrenic nerve, the brain, and how it all kind of controls what's happening. And you'll see there that if the phrenic nerve doesn't work well, then the diaphragm will kind of not be able to function. But the phrenic nerve is one of the only motor neurons you have that functions intrinsically just on a rhythm. And so it can get kind of short-circuited, so to speak, because of something like that. Somebody else asked me about hiccups, and so we'll talk about hiccups too, and why those come about or what is happening in that particular case. Okay, any other questions? So you can measure respiratory function in terms of volumes. Now, certainly we can measure respiratory function in terms of gases, right? People who work in an ICU, when somebody is experiencing respiratory failures, they will always constantly be checking blood gas concentrations from arteries, okay? But what about not doing it that way? Instead of looking at gases and what's happening to your blood in terms of oxygen and CO2 and pH, can you measure functionality on volumes, right? Because to be quite honest, pulling arterial blood is somewhat invasive. It's not horribly invasive, but it's certainly a lot more invasive than just getting blood drawn from your veins. So if you can look at lung function in terms of total volumes, that's a little bit better. And many of you have maybe done this. If you've gone into your doctor's office and they give you this kind of plastic box that has a cylinder in it, and it has usually a plastic tube coming out, and they tell you to take as hard of a breath as you can, and kind of this whole piston rises and gives a number that your doctor then uses to determine how healthy your lungs are. That is essentially what spirometry is, okay? Spirometry utilizes tools to measure lung volumes. And there really is no limit to which lung volume you can measure using spirometry. It's a pretty simple setup where you have, this is kind of the oldest type you're going to find. If you guys are taking phys lab, that's not what you're using. You guys are using a pretty well-designed apparatus that takes a force transducer that's kind of built into these different parts that you have, transfers it through your power lab, the little box that sits on the desktop, and then transitions it to the computer. But you should get a trace that looks almost exactly like this one, okay? And so that trace there of spirometry is measuring the volume of breath in and then the volume of breath out. And each breath can be defined as a tidal volume. So tidal volume is just one breath, how many mils was it? And it can be done on the inspiration as well as the expiration. So this right here is a tidal volume, and this down here is a tidal volume, okay? Usually matched, but not always. Sometimes you might take a deeper breath in than you do out, or vice versa. But so that's why tidal volume is a very individual measure. Tidal volume, like stroke volume, isn't necessarily the best measure of how much media or air is moving over time, because it's only telling you one breath. So again, in one breath, you could take a really shallow breath. On another breath, you could take a super deep one, and that's really not telling you how much air your lungs are getting. So instead, you use a measure called minute ventilation. And minute ventilation gives you a more appropriate number to work with, like cardiac output, which is telling you how much air are those lungs exchanging with the atmosphere per minute. So you kind of get a much better idea. Am I getting enough air into the lungs to provide the oxygen that I need and to provide the removal of CO2 that I need? That's much better calculated using that minute ventilation. So in order to do that, you not only need your tidal volume, but you need breathing rate as well, okay? Because then what you're going to do with tidal volume being liters per breath, you'll multiply that by your breaths per minute, and it'll give you a liters per minute, okay? Certainly a number that can change quite a bit depending on what you're doing, just like cardiac output. Stroke volume can potentially change, heart rate changes, therefore those both change cardiac output. Tidal breath or tidal volume can change, breathing rate can change, and both of those will then alter your overall minute ventilation. We can utilize spirometry and volume traces again to figure out how healthy the lungs are. And although this is showing a very standard spirometry trace, one that would describe what's happening during, let's say, quiet breathing, I would expect to see a trace like that if we were looking at breathing in all of you like right now. It is not showing the only things you can potentially measure. So there are other things we can look at, like maximal breaths in and out, breathing in as much as you possibly can to your capacity, breathing out as much possible air as you can, and so forth, okay? So now, when we start analyzing those different volumes, we essentially look at a volume trace, like this one here. And there are quite a few things that we can define looking at this trace. But basically, on this one figure, you have all of the possible lung volumes and capacities. Volumes are individual measurements, while capacities are combinations of two or more volumes. Okay? So we're going to go ahead and define each and every one. You'll see your general measurement here. This is on average. So 5,800 milliliters, all the way down here to 1,200 mils, and then kind of an exchange of what might be happening, so as things rise and fall and so forth. So we already discussed tidal volume, right? That's the volume of air entering or leaving the lungs during a single breath. On this trace, it is the volume highlighted in the blue bracket, so it's right in the middle, right in here, and it's the smallest volume that you see. Again, as I mentioned, roughly 500 mils is what tidal volume is, okay? That is an average estimate for most people. And tidal volume is, again, resting volumes. And so this isn't if you're exercising. This isn't if you've just taken a pretty quick walk across campus. This is just as you're sitting. When you're sleeping, it's a little lower than that. Not by much, but it is a little bit lower than that. Okay, now you know that you can take a super deep breath in, right? You're not limited to just that tidal volume. You'd be in a little bit of trouble if you were limited just to exchanging 500 mils per breath. So you can do more because you have reserves. There is an inspiratory reserve volume. That inspiratory reserve volume is every volume you can add on top of tidal volume. So you've already brought in a normal tidal volume. Anything additional or beyond that is the inspiratory reserve. So that's kind of the extras. If you want to use them, you can, but you certainly don't have to. Okay? You'll notice that inspiratory reserve will reach a point where it really doesn't keep going. So it's not an infinite amount of volume because your lungs are only so big and they only have so much room in which they can go. But again, you do have that reserve in case you want to take a deeper breath in. When you combine your inspiratory reserve volume with your tidal volume, that gives you your inspiratory capacity. Okay? So inspiratory capacity is the most amount of air you can possibly breathe in from the end of an expiratory cycle. And again, this is where it helps to kind of just breathe and pay attention to what's going on. So you just breathe out, and after you're done breathing out, you're going to breathe in. So you're going to bring in the tidal volume, and I'm going to tell you, keep breathing, keep breathing, keep breathing until you absolutely can't anymore, okay? From the start of that inspiration to the point where you can't anymore, that you just brought in your inspiratory capacity, okay? There is a reserve on the opposite end as well. There's an expiratory reserve. So just like I can breathe in a little bit more than my tidal volume, I can breathe out a little bit more than my tidal volume as well. So essentially what we have on the expiratory side, actually we'll go over here, is the expiratory reserve. So you breathe out a normal tidal volume, and after breathing out that normal tidal volume, there's a little bit extra. So that is your expiratory reserve. You can blow out air until you turn blue in the face, until you can't anymore, and there's no way the lungs will ever be empty. Okay? The lungs will never be empty. That volume that stays behind, that can never be completely expelled, is called the residual volume. Okay? Notice on this trace, it's in this green area at the very bottom. Notice there's no red lines going into it because it's not a volume you can change. It's not a volume you can utilize. It's not a volume that can be altered under normal physiological conditions. And so that residual volume exists because the lungs are adhered to the ribcage. And because of their compliance, they're already partially filled and partially stretched open. The only time that residual volume will, let's say, decrease is if your lungs collapse. Right? If the lungs collapse, then you're decreasing that residual volume because they're not adhered to the ribcage anymore. It can increase in people who have emphysema because it's harder for them to get air out, and so there's more that kind of gets left behind or stuck. And so you end up with possibilities of changing it, but it's disease states, it's trauma states, it's not normal physiological states. Okay? Good. Adding a couple more volumes together, so vital capacity. Vital capacity ends up being the total amount of air you can exchange between your lungs and the atmosphere utilizing all of your reserves. So you can find out what your vital capacity is. If you breathe out everything that you possibly can till you can no longer breathe out anymore, and then you start breathing in from that state, and you keep breathing in and in and in until you can't breathe in anymore, you're going to find out what that vital capacity is. And it's going to tell you roughly what that is. That is limited to the size of your lungs, okay? You really can't change that, as is your total lung capacity. That's not really something that can change either, because it is an addition of your vital capacity with that residual volume. So it's absolutely everything that the lungs are capable of holding. The last measurement, and this is actually a very common clinical measurement, is the functional residual capacity. So this is the amount of air left in the lungs after a tidal breath out, and it includes both your expiratory reserve plus the residual volume. So does anybody have asthma? Just out of curiosity? Okay, so asthma is an inflammation of the bronchioles, okay? It can cause wheezing, it can cause difficulty breathing, and part of the biggest issue with asthma is that you have high resistance. That high resistance is making it hard for air to move. So what they do is test the functional residual capacity of people with asthma to see how well their lungs are working. And so basically what they do is tell you take a deep breath in and then try to force air out. Now, what do you think would happen to somebody who has asthma who tries to force air out in a high resistance system compared to somebody who does not have asthma? How do you think those numbers would be different? Yes, Jess. So essentially somebody with asthma would have less air leaving because they would end up not being able to overcome some of that resistance in the bronchioles that are inflamed, making the airway narrow, whereas somebody who doesn't would be able to get rid of more air in that mechanism, right? And so it kind of helps to figure out how much resistance might that respiratory tract really be encountering that could be affecting air exchange. Jake? It depends on what you have. So asthma has a lot of different, let's say, subcategories, if you want to call it that. There are people who have exercise-induced asthma. There are people who have allergen-induced asthma, so pollens, cats, dogs, that kind of stuff. And there are people that have chronic inflammation, which really gets translated into a different kind of asthmatic process. It could be an autoimmune to a certain extent. Sometimes those people get categorized under the COPD category. Sometimes. There's more things with COPD that make it worse than asthma. And that's why some people with asthma will be prescribed things like the Advair discus, because it deals with inflammation long-term because it has steroids in it. Whereas your little albuterol inhaler, it's also prescribed to asthmatics, but it is as at the time needed, right, immediate type of effect. So there are different categories. Some people will have a chronic all-time inflammation that is dealt with things like steroids, like the Advair discus, right? Albuterol inhalers don't have steroids in them. Okay, good. So think about these volumes. Some of them are changeable, some of them are not changeable. And so as we start thinking about these volumes, we want to understand under different conditions, resting versus exercise, as we have up here, what would we expect to happen? So if I have a rest compared to exercise, which of these lung volumes do you believe is going to change the most? Okay, and then we'll talk about it together.